Welcome students, I am Adlin lecturer of Emmanuel College of Pharmacy. Today we see about electrophoresis. First we see the introduction to electrophoresis. Electrophoresis means means current or charged particles. Porous means movement. Movement of charged particle is called electrophoresis. Correct definition is electrophoresis is a movement of charged particles towards the opposite charged electrodes under the influence of electric current. That is called electrophoresis. Next, we see the paper electrophoresis. Paper electrophoresis is one of the separation technique. Mainly, it is used to separate amino acids, protein, peptides. It is based on the charge and size of the molecule. We separate the mixture of compounds. Migrations of charged particles take place through paper under the influence of current. Under the influence of current, the charged particles is migration take place. Next, electrophoresis is the separation technique. The compound are separated based upon charged particles towards the opposite charge electrode under the influence of electric field. It use the supporting medium is paper. Paper is the supporting medium we use in the paper electrophoresis. Paper does not contain charges that interfere with the separations of compound. Next we see the types of paper electrophoresis. It is based upon voltage. It is two types. One is low voltage paper electrophoresis. Second one is high voltage paper electrophoresis. First one, low voltage paper electrophoresis means a voltage is 100 to 300 voltage is applied across the electrodes. More number of samples can be separated on the single paper. Single paper we separate more number of samples at a one time. Bands are appear. Sample bands may appear. Second one, high voltage paper electrophoresis. It means up to 1000 voltage is applied across the electrodes. Sharp bands are formed, but low voltage the sample bands may only appear, but this high voltage paper electrophoresis sharp bands are obtained. More voltage is required. It is dangerous to operate. These are the types of paper electrophoresis. Next, we classify based on design of instrument. There are two types: horizontal paper electrophoresis. Second one is vertical paper electrophoresis. These are the two types of paper electrophoresis based on design of instruments. This is the picture of the horizontal paper electrophoresis. In this electrophoresis, the paper will present in the horizontal portion. Vertical paper, second one, vertical paper electrophoresis. In this electrophoresis, the paper is vertically placed. This is the picture. These are the instrumentation of the picture of the paper electrophoresis system. Next we see the working. Next we see the working of paper electrophoresis. First we select the paper. The paper is used in the paper electrophoresis is Wartsman filter paper. Thickness is 1 to 3 millimeter thickness. It first this paper is dissolved with water and dipped into HCl or EDTA solution. After drying, the paper is ready for performing the paper electrophoresis. This is the paper suitable. Next, we spotted the sample by using the capillary tube. The sample is spotted in Wartsman filter paper and dried. Next, we select the buffers. What are the buffers we use? Paper electrophoresis means acetate buffer, phosphate buffer, and barbitone buffers. Next, the buffer solutions in two rods fit with the electrodes. Now, electric field is applied across the system. The charged particles of sample migrates along the strip towards respective electrodes of opposite polarity. The homogeneous group of particles migrate as separate bands. The process is carried out for 14 to 16 hours this process is done. Next we see the staining. The staining products are used in the paper electrophoresis that are protein compound. We use the bromophenol blue in acetic acid. Nucleic acid compound means methyl green pyronine and ethidium bromides, and polysaccharide compound means iodine dye is used in the paper electrophoresis. Glycoprotein compound means alkyne blue dye is used in paper electrophoresis. This is the stainings. Next, the paper electrophoresis for amaxylin preparations. First, we prepare the paper strips. Cut high quality Wattsman number 1 filter paper into strips approximately 10 cm in length. Second step, sample applications. Spot a sample of the amaxylin solution onto the center of each paper strip. Allow the spots to dry completely. Third steps, electrophoresis setup. Place the paper strips in a horizontal electrophoresis chamber filled with an appropriate buffer solution such as phosphate buffer at pH 7.4. The next step, separation process. Run the electrophoresis for the specified time 30 to 60 minutes, allowing amaxylin to migrate based on the charge and size. Next step, visualization. After electrophoresis, remove the remove the paper strips and dry them. Stain the strips with an appropriate reagent such as ninhydrin to visualize the separate bands. Last process, analysis. Examine the sustained bands under UV light or a suitable imaging system. Measure the distance migrated by amaxylin and compare it to node. This is the steps we used in paper electrophoresis for amaxylin separations. Next we see the factors affecting electrophoresis. First one is properties of supporting medium affect the paper electrophoresis. Next, the strength of the strength of the electric field also affect electrophoresis, and the pH, temperature of the buffer, pH of the buffer also affect the paper electrophoresis. The pH depends upon the ionic strength and usage of ionic buffer. So pH affect the electrophoresis. Next, molecular mass of sample. Molecular mass, molecular weight, molecular size of the particles also affect paper electrophoresis. The molecular weight, the compound having more molecular weight, the rate of movement is slower. The compound having less molecular weight, the rate of movement is faster. So, molecular weight of particles also affect the paper electrophoresis. The molecular size and shape. The size and shape of molecule is directly proportional to movement of the charged particles. So, the molecular size and shape of the particles also affect the paper electrophoresis. Next, advantages of paper electrophoresis. First point is it is simple and it is inexpensive process. Third one, easily available. Fourth advantage, easy to handle the instruments. Disadvantage of paper electrophoresis means certain compounds like protein, hydrophilic molecule cannot be resolved because of adsorptive and property of paper. It results in tailing compound bands. Third point, electroosmotic flow may occur. Sometimes they occur the electroosmotic flow. The fourth point, time taking process. It is a time consuming process. It take almost 14 to 16 hours for separation process. So it is the time taking process. This is the advantage and disadvantages of paper electrophoresis. Next one, applications of paper electrophoresis. First application is it is used for separations and identification of protein. Protein will be separated and identified by using the paper electrophoresis. Second application, it is used for separation and identifications of amino acids and micronutrients also separate and identify by using the paper electrophoresis. It is also used for separation and identifications of vitamins, minerals, and radioactive substances. These also separated and identified by using the paper electrophoresis. And next uses is it is used for separation and separations of fluorescence producing pharmaceutical compounds. The fluorescence also separated by using the paper electrophoresis. Next uses is serum analysis for diagnostic purpose also use separations of amino acid protein in serum. Third one, separation on enzyme in blood, separations of antibiotics in different samples. Next, the muscle proteins, egg white protein, and snake insect venom analysis. These are done by paper electrophoresis. The muscle protein, egg white protein, and snake insect venom analysis. These done by this paper electrophoresis. Used in the separation and identification of alkalis. Alkalis also identified and separated by using the paper electrophoresis technique. It can also be used to test the suitability of municipal water supplies, the toxicity of water, and other environmental components. These are the applications of paper electrophoresis. This day we see the introduction about electrophoresis. Um, Next, we see the introductions of paper electrophoresis. As this, we see the types of paper electrophoresis. It is based on voltage applied to types: low voltage paper electrophoresis, high voltage paper electrophoresis. Next, we classify based upon the design of instrument. These are classified into two: horizontal paper electrophoresis and vertical paper electrophoresis. Their structures are C. Next, we see the working of the paper electrophoresis. Next, staining compounds, and the next we see the paper electrophoresis for amaxylin separations. That also see. We see the factors affecting electrophoresis. What are the factors affecting means properties of supporting medium, strength of electric field, temperature of buffer, pH of buffer, molecular mass of sample, net charge on the particle, shape and size of the particle. These are the factors affecting the paper electrophoresis. Next we see the advantages and disadvantages of paper electrophoresis. Next we see the applications of paper electrophoresis. Uh, these are the paper electrophoresis techniques. Thank you.